This is a paper that looked at the limited detection of 26 different COVID tests in both Australia and Canada. So why is this relevant? Well, to date, there's over 1,000 different COVID tests that are commercially available for rapid testing, where most of these detect the nucleocapsid protein, which is relevant in this study. And it's relevant because in this study, they used two nucleocapsid protein reference materials to find the limited detection of each of these 26 COVID rapid tests. Additionally, they tested two of the COVID-19 influenza A/B combined rapid antigen tests, which is good to see how they compare and stack up to the COVID-specific tests. So, as a quick reminder of how these tests actually work, what they do is they exclusively capture and detect free nucleocapsid proteins that are released from infected cells and viruses upon lysis, meaning that if there are COVID-infected cells or free COVID viruses that are captured on your cotton tip swab, when those are split apart in the stirring process, cell lysis, breaking apart, the inner protein is then released from the COVID virus, which is the nucleocapsid protein, which is then detected by the rapid antigen test using fluorescence typically. And fluorescence is just that pink line that you see. And this comparative study was done in Q1 of 2023, so early in the year of 2023, making this study pretty relevant to right now in 2024. Now, in their comparative analysis, the rapid antigen tests were assessed at 0.1 nanomoles, followed by an additional assessment at 0.05 nanomoles if the tests were positive on that 0.1 nanomoles. And if the tests were negative at 0.1 nanomoles, they increased the concentration to 1 nanomole. And they did this until they got a negative on a test or a positive on a test, whichever way the testing went. So, for example, this was the testing that was performed. So, for instance, the tests that are having a positive on 0.05 nanomoles are going to be the most sensitive tests, while the tests that are first testing positive at 1 nanomoles are going to be the least sensitive tests. And as shown here, a faint positive is still a positive, which means you're still infectious with COVID. And for reference, the top 6 tests here in no particular order are BTNX cassette, FANT test, FlowFlex, EnoScreen, JustCheck, Meds Up. So if you have these options and selections of rapid antigen tests, these are the ones that you're going to want to select. And this chart right here demonstrates why not all rapid antigen tests are the same. So for instance, this is FAN test, which is very sensitive at a 0.05 nanomole concentration of COVID, of the nucleocapsid protein of COVID to be specific. So if I hold very still, you can see a light positive line right there, and then it gets darker and then even darker still. But for less sensitive tests like these ones down here, so for instance, one of these ones right here, like MP Bio, that just starts detecting at a 0.1 nanometer concentration, that one will barely detect the COVID at the same concentration as the FAN test. You can see the line is much lighter. And we see the same thing over here at the higher concentration, which would be selecting one of these ones down here, like BTNX, where despite there being a high COVID concentration with the nucleocapsid protein, where the FAN test is having a very strong band, this other test would have a very light band, which is why it's very important to pick a high quality, high sensitivity rapid antigen test. I prefer FlowFlex personally, but any of these would be great. And then here's information for the 3-in-1 test. This would be a FAN test 3-in-1 with COVID and the flu, and then Touch Bio for COVID and the flu. You can see out of these two options, FAN test would be the better one. It has a sensitivity down to 0.1 nanomole of the nucleocapsid protein of COVID, while on Touch Bio at the same concentration, it's showing a negative result. And for the flu A test that was run, it seems to be pretty equivalent, where you're having a decently strong band at the 5 nanomole concentration, you're having a lighter band at the 1 nanomole, and then they're both negative at the 0.1 nanomole.